Welcome back, everyone, for another deep dive. And today we're taking a look at a recent Western Equine Encephalitis Virus outbreak in Argentina.
Oh, wow.
I know you're particularly interested in neurology, so I think you'll find this research article from Virus's journal pretty fascinating.
Yeah, for
They
sure.
go into great detail about the clinical and pathological findings.
I'm ready.
And this outbreak is particularly interesting because it comes after a nearly 40-year quiet period, and it may involve a new DEE lineage.
It is definitely significant. The article reports 1530 suspected and confirmed equine cases.
Wow.
Yeah, across 17 provinces since late November 2023. And even more alarming is the jump in human cases, 73 confirmed infections and 7 fatalities in both Argentina and Uruguay.
Yeah, and adding to that, the article even suggests that the actual number of cases could be even higher.
Right.
Due to underreporting, especially in those rural areas where access to diagnostic testing is limited.
Yeah, that's right. Underreporting always an issue with outbreaks, especially in areas with limited resources. It really skews our understanding of the true impact and spread of the virus.
Absolutely. So the researchers investigated 34 horses that were presenting with neurological symptoms, and they were able to detect WEEV RNA in 74% of them.
Pretty high percentage.
Yeah, using a combination of RT-PCR, real-time RT-qPCR, and virus isolation in Vero E6 cells.
Interesting.
So they primarily focused on brain tissue samples, and this is where it gets kind of interesting. Despite confirmed brain infections in many horses, WEEV RNA was not detected in their cerebrospinal fluid.
That is curious. You would typically expect to see viral presence in the cerebrospinal fluid with encephalitis.
Right. It definitely raises questions about the virus's neurotropism and its behavior in this outbreak.
Absolutely. It challenges some of the conventional understandings of how WED affects the central nervous system. And to add to the intrigue, they also detected WEEV in the brain of a sheep presenting with neurological symptoms.
Oh, well.
Yeah, which raises concerns about potential cross-species transmission and a broader host range than we previously understood.
That's certainly concerning. This is where the phylogenetic analysis comes in.
Right.
They analyzed partial NSP4 gene sequences from 3 isolated WEEV strains to understand their evolutionary relationships.
And what they found is quite compelling. The isolates clustered closely with recent strains from Brazil and Uruguay, forming a distinct clade, potentially representing a novel lineage. They're calling it Lineage C.
So we're potentially dealing with a new player on the field here.
Exactly. And the article suggests that this novel lineage may be driving this widespread outbreak in South America.
So we have this potentially new lineage spreading rapidly and displaying some unusual characteristics in terms of its presence in the central nervous system.
Precisely.
So to understand the extent of exposure and immune response, the researchers conducted serological testing using the virus neutralization test, or VNT, on 50 horses.
Okay.
What did those results reveal?
Well, they found a surprisingly high seroprevalence. 73.3% of the horses that were in contact with infected individuals had WEEV antibodies.
Wow.
Yeah, even if they weren't showing any clinical signs.
So that signifies a large number of asymptomatic carriers potentially contributing to the silent spread of the virus.
Exactly. And it highlights the need for comprehensive surveillance and control measures, considering that seemingly healthy animals could still be spreading the virus.
Absolutely. They also looked at antibody titers. Can you elaborate on those findings?
Sure. They observed a correlation between antibody levels and disease severity. The horses with confirmed WEEV and those exhibiting neurological signs had the highest antibody titers. Interestingly, antibody titers in 4 recovered horses gradually declined over a 3-month monitoring period.
So that raises questions about the longevity of immunity after natural infection, which has implications for vaccination strategies.
Absolutely.
Oh.
And a challenge they highlighted was the inability of current serological tests to differentiate natural infection from vaccine-induced immunity. This points to a crucial area for improvement in diagnostic tools.
For sure. Now let's move on to the pathological findings. The researchers examined 6 horse brains, revealing some telling details.
Okay.
What did they find?
The examination showed severe meningoencephalitis in all 6 brains, with a significant neutrophilic infiltration. This inflammation was particularly intense in the internal capsule of thalamus and brainstem. They even observed some perivascular cuffing, which suggests inflammation extending beyond the brain parenchyma.
To pinpoint where the virus was replicating? They utilized the technique called RNA-scope in situ hybridization.
Yes, this technique allowed them to visualize the distribution of viral RNA within the brain tissue.
And what did that reveal?
Interestingly, they found WEEV RNA in various cell types within the brain parenchyma, including neurons, glial cells, and inflammatory cells, but notably not in the perivascular inflammatory cells.
So that's a unique finding that adds another layer of complexity to our understanding of how this particular WEE lineage behaves.
Indeed. They also found a positive correlation between the abundance of viral RNA detected through RNA-scope ISH, the RT-qPCR results, and the severity of the inflammatory response. This reinforces the link between viral replication inflammation and the progression of the neurological disease.
And where was the viral load the highest?
The brainstem and thalamus had the highest viral load, which makes these regions critical for diagnostic sampling. These findings also suggest that certain areas of the brain might be more susceptible to this particular VETV lineage.
Okay, so we've covered a lot of ground. We've got this potential new WEEV lineage circulating, causing severe neurological disease in horses and displaying some unusual characteristics like its absence in cerebrospinal fluid despite brain involvement.
Precisely. And the high seroprevalence suggests a significant level of silent spread, further complicating control efforts.
This raises several important questions. How did this new lineage emerge? What factors are contributing to its rapid spread? And what are the long-term implications for both animal and human health?
That's the crux of the matter, isn't it? Well, the article actually presents some fascinating data in a table outlining the clinical signs observed in the horses.
I'd love to hear it.
They break down the signs into categories: general behavior, cranial nerves gait, and posture and sensorium.
Okay.
In the general category, fever was the most frequently reported sign. Ataxia, or incoordination, was the predominant finding under gait and posture, with 79.4% of the horses displaying this sign.
That makes sense considering the virus's impact on the cerebellum and brainstem.
Exactly. The next most common signs in that category were paresis, or weakness, and hypermetria, exaggerated movements.
Mm-hmm.
In the behavior category, depression was the most common, followed by somnolence and head pressing.
Head pressing is a classic sign of forebrain involvement, isn't it?
It is, and it aligns with their pathological findings of inflammation in areas like the thalamus.
So we're seeing a mix of cerebellar brainstem and forebrain signs, painting a picture of widespread neurological involvement.
Precisely. Now, moving on to cranial nerve deficits. They noted facial nerve paralysis in 35.3% of the horses, followed by changes in menace response and tongue paralysis.
Interesting. And what about alterations in sensorium?
They observed signs of obtundation or reduced responsiveness in 26.5% of the horses. And in some severe cases, seizures were also reported.
This clinical picture paints a pretty grim picture, doesn't it?
It does. It really highlights the severity of the neurological disease caused by this particular WEEV lineage.
Now let's delve into the nitty-gritty of the pathological findings. The article provides detailed descriptions of the microscopic changes observed in the horse brains.
Right.
What did they do?
They used hematoxylin and eosin staining for general examination and immunohistochemistry to specifically detect WEEV antigen.
And what did those analyses reveal?
The microscopic examination revealed widespread neuronal degeneration and necrosis, accompanied by marked infiltration of inflammatory cells. Neutrophils were the predominant cell type, indicating an acute inflammatory response.
Where was this inflammation most prominent?
The lesions were most severe in the gray matter of the thalamus, basal ganglia, brainstem, and cerebellum. They even observed some perivascular cuffing, which suggests inflammation extending beyond the brain parenchyma.
The article also mentions using immunohistochemistry to detect viral antigens. What did that show?
Immunohistochemistry highlighted the presence of WEEV antigen within neurons, glial cells, and inflammatory cells. This confirms the direct infection of these cell types by the virus.
Did they observe any differences in antigen distribution between brain regions?
Interestingly, they found that the distribution of WEEV antigen varied depending on the brain area. In the cerebrum, the antigen was primarily localized in the neurons of the cortex and basal ganglia. In contrast, in the cerebellum, the antigen was predominantly found in the Purkinje cells and the molecular layer.
That's fascinating. It suggests that this WEEV lineage might have selective tropism for different neuronal populations within the brain.
Absolutely. And these findings provide valuable insights into the pathogenesis of WEEV infection and how it causes such severe neurological damage.
The article also includes several figures illustrating the pathological findings. One figure particularly caught my eye. It shows immunohistochemical staining of the brainstem, highlighting WEEV antigen in neurons and inflammatory cells.
Yes, That figure really vividly demonstrates the extent of viral infection within the brainstem, a critical area for vital functions.
And another figure shows a high-resolution image of a Purkinje shell in the cerebellum with WEEV antigen clearly visible within the cytoplasm.
Powerful image. It underscores the vulnerability of Purkinje cells to WEEV infection, which could explain the ataxia and other cerebellar signs observed in the horses.
Okay, so we've discussed the clinical signs, the microscopic changes, and the immunohistochemical findings. It's clear that this WEEV lineage is causing widespread and severe neurological damage in horses.
Precisely. And the details presented in the figures and tables really bring those findings to life, wouldn't you say?
Absolutely.
Yeah.
Now, given all this information, what can we glean about the virus's unique behavior and the factors contributing to its spread?
That's the crux of the matter, isn't it?
So what does the article say about those factors?
Well, the article suggests that several factors might be at play. The decline in herd immunity due to the long absence of WEEV likely created a susceptible population ripe for an outbreak.
That's a good point. Without prior exposure, those horses lack the protective antibodies to combat the virus, effectively allowing for rapid transmission. What about the role of environmental factors?
Right. The article points to potential changes in mosquito vector populations and their distribution. Variations in temperature and rainfall patterns could create favorable conditions for mosquito breeding and survival, leading to increased virus transmission.
So we have a combination of a susceptible horse population and potential environmental changes contributing to this outbreak.
Precisely. The article also raises the possibility that this novel lineage C might have enhanced virulence or transmissibility compared to previous strains.
Now that's a concerning thought. Is there any evidence to support that theory?
Well, while more research is needed, the high morbidity and mortality rates observed in this outbreak, along with the virus's unique characteristics, warrant further investigation into its virulence and transmission dynamics.
So we're dealing with a complex interplay of factors that have led to this significant outbreak.
Absolutely. And the article stresses the need for ongoing surveillance vector control measures and public awareness campaigns to mitigate the spread of WEEV.
What about vaccination?
Vaccination is undoubtedly a crucial tool in controlling WEEV. However, the article highlights the challenge posed by the potential new lineage and the need for vaccines that provide adequate protection against this specific strain.
It seems like we're in a race against a constantly evolving virus.
Indeed, the article concludes by emphasizing the importance of continued research to understand the evolution of WEEV, its pathogenesis, and the development of effective vaccines and treatment strategies.
Okay, so for all of you listening, the key takeaways from this deep dive are we're dealing with a potentially new WEEV lineage causing a significant outbreak in South America. This lineage displays some unusual characteristics, including its absence in cerebrospinal fluid, despite confirmed encephalitis. Several factors likely contributed to this outbreak, including a decline in herd immunity, potential environmental changes, and the possibility of enhanced virulence of this new strain. And continued research and surveillance are crucial to stay ahead of this evolving threat.
Absolutely. And this outbreak serves as a stark reminder for neurologists like yourself to remain vigilant, stay informed about emerging infectious diseases, and consider WEEV as a potential diagnosis in horses presenting with neurological signs.
This has been a fascinating and in-depth exploration of a pressing issue. What are your final thoughts for everyone listening?
You know, the current outbreak really underscores the interconnectedness of animal and human health. It serves as a potent reminder that we need to remain proactive in our efforts to understand, prevent, and control infectious diseases. The burning question remains, what does the future hold for WEE in South America and beyond? Only time will tell, but continued research and collaboration will be key to navigating this evolving landscape.
From Vet NeuroJar, keep those minds inspired, hearts light, and tails wagging.